guys, we are starting the main cycle, that is urea cycle. So we have discussed the ammonia has reached our liver. So we are starting urea cycle. This urea cycle is also known as ornithine cycle. Ornithine cycle or Krebs, not Krebs cycle, Krebs-Hansley cycle. Okay. So these are the other name of urea cycle. In urea cycle, what we have to know first is where it is taking place. That is in liver, right? It is taking place in liver. In liver, both mitochondria and cytoplasm. Normally, in biochemistry, many reaction occurs or many cycle occurs in one of the organelle. In cytoplasm, nucleus or mitochondria. But here, urea cycle is taking place both in cytoplasm and mitochondria. So you have to remember three main important processes which are taking place both in mitochondria and cytoplasm. One is our urea cycle. The other is heme synthesis. Then other is gluconeogenesis. You can just remember a mnemonics HUG. H for heme synthesis, U for our urea cycle, and G for gluconeogenesis. So just give a hug, right? To relax for someone. So hug. So these all takes place both in cytoplasm and cytoplasm. These three reactions. Just remember, many MCQ will come. So we will continue with our urea cycle or ornithine cycle or Krebs-Hansley cycle. Okay? Our urea cycle. We know that now the ammonia is in our mitochondria. Very easy cycle. Now the ammonia will combine with carbon dioxide. Okay? That will form carbonyl phosphate. Okay? Will form carbonyl phosphate. So this is taking place inside our mitochondria. This is our mitochondria. And here in the lower we have our cytoplasm. Here is our mitochondria and this is our cytoplasm. Now this carbonyl phosphate is formed by an enzyme known as carbonyl phosphate synthetase and 1. Synthetase 1. Why tase? Because we are using energy here. In biochemistry, wherever you use energy, then the enzyme will be synthetase, not synthase. Carbonyl phosphate synthetase 1. Okay. Now, next step is the ornithine, which is already present in where? Cytoplasm. Ornithine is already present in cytoplasm, will be transported to the mitochondria by a transporter, ornithine transporter 1G. Okay. So now there is an enzyme present in mitochondria. The enzyme name is, so you can divide mitochondria and cytoplasm like this: ornithine transcarboxylase. Ornithine transcarboxylase. This ornithine and carbonyl phosphate, in the presence of ornithine transcarboxylase, they will form a product which is known as citrulline. Okay, they will form citrulline. Okay, now citrulline will be converted to arginosuccinate. Arginosuccinate. Here also the enzyme will be arginosuccinate synthetase. Why synthetase? Here also we are using energy. Here energy is like the ATP is becoming AMP. Okay, ATP is becoming AMP. In this step, one guest is coming here. One guest is entering here. That is our aspartate. So how arginosuccinate forming? Citrulline they will combine with aspartate. They will form arginosuccinate. Okay. Now arginosuccinate will be converted to fumarate. Fumarate will be going outside cycle and arginine. The enzyme is arginosuccinase. No Mg is here. That is arginosuccinase. Now arginine will be acted by an enzyme, that is our arginase. Okay, that is arginase. and they will form, one is our ornithine and other is what? Our urea, the byproduct. This is our cycle, right? Urea will be formed. So it will go to kidney and be excreted. Here water is also taking place. Water is also taking place. Arginine will be converted to urea and ornithine. So this is very simple. This is our Krebs cycle. Now what we have to listen, carefully listen here is, what are the mitochondrial enzymes? See, ornithine transcarboxylase. See, we have one more enzyme here. It is known as NAG. You know snake, right? Hindi mein NAG kya hai? Snake hai, NAG. N-acetyl glutamate. This N-acetyl glutamate will activate carboxyl phosphate synthetase 1. Okay? So NAG is N-acetyl glutamate. What is the function? They activate carboxyl synthesis 1. So NAG and ornithine transcarboxylase are in mitochondria. Other enzyme, arginine synthetase, arginase, all the enzymes starting with A, you can remember like that. They are in cytoplasm. Clear? Now how many? See, 2 ATP is becoming 2 ADP here. We are losing 1 phosphorus here, right? 1 phosphorus is lost here. Now here ATP is becoming AMP. We are losing 2 phosphorus here. Okay? From 1 ATP we are losing 1 phosphorus. So total we are losing 1 phosphorus plus 1 phosphorus. 2 phosphorus is lost here. Also 2 phosphorus is lost here. How many ATP we are using here? 2 ATP. How many ATP here? 1 ATP. So total energetics will be, we are using 3 ATP. How 3 ATP? 2 ATP here, 1 ATP here. And how many phosphorus? From 1 ATP to 1 ATP we are losing 1 phosphorus. Now other second ATP we are using again 1 phosphorus. So 2 phosphorus lost here and 1 ATP to adenosine monophosphate. That is we are losing 2 phosphorus. So total 2 plus 2, that is 4 phosphorus is lost. 4 phosphorus is lost. This is the energetics. Okay. Now next thing you have to remember here is the importance of enzyme. Okay. Now arginosuccinase, it is a type of enzyme under lyase. Okay, it comes under lyase class. You see here arginase. Okay, but arginase is in the presence of water, so it comes under the enzyme hydroxylase. Okay, it comes under the enzyme hydroxylase. Now, so these all are about the main things. Okay, main things in urea cycle. Now we have to see some disorders, what will happen. So it is very easy, right? I'm repeating once more. Ammonia and water, so carbon dioxide will form carbamyl phosphate in the presence of carbamyl phosphate synthetase 1. So the synthetase means they are using energy. 2 ATP will become 2 ATP. Ornithine already present in the cytoplasm will be transported to the mitochondria by the gene ornithine transporter 1. Okay, the gene is ornithine transporter 1. The enzyme is ornithine transcarboxylase. They will form what? Citrulline. This citrulline will be acted by the enzyme arginosuccinate synthetase. We are synthesizing arginosuccinate. So arginosuccinate synthetase. So here also we are using energy. Okay. Here a guest is entering, that is our aspartate. In this chain aspartate is entering. So citrulline plus aspartate, they will form arginosuccinate. This arginosuccinate is lyased, break by arginosuccinase to form fumarate and arginine. This fumarate will go to Krebs cycle. Then arginine will be acted by arginase in the presence of water to form our urea. This is our byproduct. It will go to the kidney and excreted and ornithine. Okay. Now one more thing you have to remember here. What is the formula of urea? That is NH2CO NH2. Right. So the MCQ will be one amino group is from our urea. Now in urea we have 2 amino group. One will be from first amino group from ammonia. Okay. The second one is from where? Is from our guest, that is our aspartate. Okay. That is our aspartate. Clear? Now we will study some disorder due to ammonia. This urea cycle. That is, we have one disease known as, I think all you are clear. I am discussing the disease also here. If carbamyl phosphate synthetase 1, this enzyme Okay, this carbamyl phosphate synthetase 1. If CP1 is absent or deficient, it is called as hyperammonemia type 1. Hyperammonemia type 1. Now, if this enzyme ornithine transcarboxylase is absent, ornithine transcarboxylase, that is this enzyme here, if this is absent, it will cause hyperammonemia. Hyperammonemia means increase in ammonia, right? If this is doesn't occur, the ammonia can be detoxified to a less toxic urea. It is hyperammonemia type 2. This hyperammonia type 2 is X-linked partly dominant. Very important. X-linked partly dominant. Okay, X-linked partly dominant. Now we have one more very important disease. It is known as triple H syndrome. Okay, triple H syndrome means it's not the WWO triple H. It is triple H in biochemistry. Next disease is triple H syndrome. You can call it as triple H. Okay, so what are the triple H here? Okay, here first H is for hyperammonemia. Second, if hyperammonemia will be here. Second is hyperornithinemia. Ornithine will be increased. Then third one is homocitrullinemia. So the triplet syndrome is what? Hyperammonemia, hyperornithemia and homocitrullinemia. What is the reason for this? This is not the deficiency of any enzyme. This is the mutation in our mutation of ornithine transport gene, gene 1. See, I have told you ornithine will be transported to mitochondria by ornithine transport gene. So if there is any mutation occurs, triple H syndrome will come. That is hyperammonemia, hyperornithemia and homocitrullinemia. So deficiency of carbamyl phosphate synthetase 1 will cause hyperammonemia type 1. Ornithine transcarboxylase enzyme deficiency cause hyperammonemia type 2. This X-linked partly dominant. Normally in biochemistry, all enzyme deficiency diseases are autosomal recessive. So some rare diseases are X-linked, okay, X-linked dominant. So hyperammonemia type 2 is X-linked dominant. It is asked in many exams. Then third one is triple H syndrome. It is close to mutation of ornithine transport gene 1. Hyperammonemia, hyperornithemia and homocitrullinemia. Clear, right? So these are all about urea cycle. So urea cycle takes place in the liver. This is very, very easy. All the enzymes are clear, right? Don't remember NAG. This NAG is known as 6th enzyme of urea cycle. So this is also an enzyme, 6th enzyme of urea cycle. What is the function of NAG? It will activate carbamyl phosphate synthetase 1. Okay, so this is called as 6. Energy we know 3 ATP and we are losing 4 phosphate. Clear? So these are the all important things about urea cycle. So we are done with urea cycle.